The structure of DNA is a double helix. It is composed of two sugar phosphate backbones. The product that connects the backbones, they're called base pairs, composed of pyrimidines and pyridines. They will be discussed later on. The bases of DNA, the pyrimidines and the purines, are the four main components of DNA. You have adenine, guanine, thymine, and cytosine. These base pairs will pair together in order to create the ladder that you see when you think of DNA. The structure of DNA runs anti-parallel to each other, meaning that the two phosphate backbones do not run in the same direction. You have a 5' end and a 3' end. One side runs from 5 to 3, and then the complementary side runs from the 3 to the 5' end. As you can see, adenine binds with thymine, and cytosine binds with guanine, the base pairs that were just previously discussed. As you can see here, we have a sequence of DNA nucleotides. Again, these are the bases, adenine, guanine, thymine, and cytosine, that compose the backbone of DNA. Here is a table used to decipher which amino acid your nucleotide bases are coding for. A codon is a sequence of 3 bases that correspond to a specific amino acid. Each amino acid will have a codon, but they can also have multiple codons associated with a specific amino acid. Some codons, as you can see outlined in red, are stop codons. This means that the DNA should no longer continue with its process. Here you can see a summary of DNA replication. Our first step is number 1 over to the left. The helicase, which is an enzyme, unwinds the parental double helix. Step 2, the single-strand binding proteins stabilize the unwound parental DNA. Next, the leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. Then the lagging strand is synthesized discontinuously. Primase, another enzyme, synthesizes a short RNA primer which is extended by DNA polymerase to form an Okazaki fragment. After the RNA primer is replaced by DNA by another DNA polymerase that is not shown in this picture, DNA ligase acts like a glue to join the Okazaki fragment into the growing strand. Here you can see transcription occurring. It happens after replication. Transcription uses the strand of complementary mRNA made during replication in order to continue to make new strands of DNA. You may ask what happens in the case where a mutation or something affects the DNA to where it's not properly made. Well, here you can see that there is a mechanism in order to prevent that mutation from being passed on. A thymine dimer here, which is a type of mutation, distorts the DNA molecule. Again, you have an enzyme called a nuclease that cuts the damaged DNA strand at 2 points, and then it is removed. In step 3, you can see the light blue section of DNA that is the correct portion of DNA that is made by repair synthesis of DNA polymerase. It fills in the missing nucleotides. Next, we have DNA ligase again that acts as a glue and seals the free end of the new DNA to the old DNA, making the strand complete. Translation is the third step. Here you see translation. It is the third step after transcription. In translation, messenger RNA produced by transcription is decoded by the ribosome to produce a specific amino acid chain or a polypeptide. It will later fold into an active protein. In humans, translation occurs across the membrane of the endoplasmic reticulum. Translation proceeds in 4 phases: activation, initiation, elongation, and termination. In activation, the correct amino acid is covalently bonded to the correct transfer RNA, or tRNA. Initiation involves small subunits of the ribosome that bind to the 5' end of the mRNA with help of initiation factors. Elongation then begins by the appropriate amino acid tRNA binding to the codon in the A site of the ribosome. Once a stop codon is reached, a release factor reads the triplet or the codon, and polypeptide synthesis ends. The polypeptide is then released from the tRNA. The tRNA is released into the ribosome, and the 2 ribosomal subunits separate from the mRNA. Here you see 2 chromosomes, an X and a Y chromosome. A chromosome is an organized structure of DNA and protein found in all cells. It is a single piece of coiled DNA containing many genes, regulatory elements, and other nucleotide sequences. Chromosomes also contain DNA-bound proteins which serve to package DNA and control its functions. The larger chromosome seen toward the top of the picture is the X chromosome. The smaller one at the bottom is the Y chromosome.